This is part 1, Model Building and Presentation of Protein Synthesis by Anthony Travato. So what is meant by protein synthesis? Protein synthesis is essentially the process of making proteins. Proteins are the functional unit made of one or more polypeptides, which are chains of amino acids. There are different types of proteins which serve different functions, which include structural, regulating metabolic function, cell communication, sensory proteins, and storage and transport. But how do these proteins get made and get the genetic information in which they need? Central dogma describes the molecular biology of the directional process of a gene that codes a protein, which is expressed from DNA to RNA to protein. This is done in 2 stages, transcription and translation, each containing many processes in between. Transcription is the process of producing messenger RNA, or mRNA, from a specific strand of DNA known as a gene, to then carry the genetic information to be translated and turned into a protein. So what is RNA? RNA, or ribonucleic acid, is often a single-stranded molecule containing a backbone consisting of an alternating ribose sugar and a phosphate group, rather than the deoxyribose sugar found in the double-stranded DNA. RNA also contains the nitrogenous base uracil instead of thymine, which bonds with adenine. Template and non-template strands. DNA is double-stranded, with one strand being the template strand and the other being the non-template strand. The template strand is the strand that serves as the template for the messenger RNA molecules, or mRNA, to be synthesized and moves in a 3' to 5' direction. The non-template strand is the other strand in DNA that is incapable of serving as the template strand for the mRNA molecules to be synthesized due to its 5' to 3' direction. The reason why the 5' to 3' strand is incapable for serving as the template strand for mRNA synthesis is because mRNA is synthesized in a 5' to 3' direction, which needs to be synthesized from a complementary strand, which is the 3' to 5' template strand. Now, what does 3' and 5' actually mean? Well, deoxyribonucleic acid, or DNA, consists of a double helix sugar phosphate backbone, which is composed of a deoxyribose sugar and a phosphate group. Deoxyribose sugar consists of 5 carbon atoms, and the phosphate group will either connect to the 3rd or 5th carbon atom, referred to as 5' and 3'. If we take a look at the deoxyribose sugar molecule, you can see that the carbon atoms in the sugar are labeled 1 to 5 in order in a clockwise direction. The template strand moves from 3' to 5', while the non-template strand complements this, moving from 5' to 3'. This means that the template strand in the first phosphate group binds to the 3rd carbon in the deoxyribose sugar molecule and ends with the phosphate group binding to the 5th carbon atom in the deoxyribose sugar molecule. For the non-template strand, the first phosphate group is connected to the 5th carbon atom and ends with the phosphate group binding to the 3rd carbon in the deoxyribose sugar molecule. In the transcription phase, when RNA is synthesized, messenger RNA, or mRNA, is synthesized from 5' to 3' as an enzyme called RNA polymerase uses the template strand to synthesize a complementary sequence of the nitrogenous bases in the mRNA strand. RNA polymerase is an essential enzyme in the transcription process, which is responsible for synthesizing new strands of RNA using ribonucleotides rather than deoxyribonucleotides. Deoxyribonucleotides include adenine, thymine, guanine, and cytosine, in which adenine only bonds with thymine and guanine only bonds with cytosine using hydrogen bonds. Ribonucleotides are very similar to deoxyribonucleotides, except it has a ribose sugar molecule instead of a deoxyribose sugar molecule, and uses the nitrogenous base uracil instead of thymine. It begins with the initiation phase by binding to a specific point in the gene known as the promoter, which is located at the start of the gene, and separates the DNA strands into the template and non-template strand in the 3' to 5' direction. The template strand then acts as the template for RNA polymerase to build RNA molecules out of complementary ribonucleotides being read from 5' to 3' in a process called elongation. It then goes through the process called termination, where a sequence of DNA codes for a signal to RNA polymerase to terminate transcription as the transcription process is complete. In eukaryotes, mRNA needs to undergo extra processing before it can begin translation. mRNA consists of introns and exons, where introns are the parts that will be removed from the mRNA and exons are the parts that will be stuck back together, giving the final mRNA. This process is called splicing and is shown in this model. The introns are removed from the pre-mRNA and leave behind the exons for the final mRNA to be translated. Translation is the process that turns the information in the mRNA into a series of amino acids to build protein. The mRNA leaves the nucleus, moves into the cytoplasm, and enters the ribosome. Ribosomes are composed of ribosomal RNA, or rRNA, and protein, which help translate the information from mRNA. How is mRNA actually translated? The ribonucleotides in mRNA are grouped in a triplet sequence known as codons, which code for a specific amino acid. Transfer RNA, or tRNA, contain a specific amino acid as well as an anticodon, which is a 3-nucleotide sequence that binds to complementary codons. Amino acids are molecules that combine to form proteins. Every codon sequence codes for a specific amino acid. Each amino acid is coded from specific codon sequences as seen in the diagram on the screen. As seen in the diagram before, specific amino acids code for start and stop amino acids. Methionine, MET, is coded from the codon sequence AUG and starts the process of adding amino acids growing the polypeptide chains of amino acids, whereas the codon sequence UAA, UAG, and UGA code to stop amino acids from being added to the polypeptide chain. How this works is the tRNA molecule matches their anticodon sequence to the codon corresponding sequence, starting with methionine. The next codon is matched with the corresponding anticodon tRNA molecule, with the next amino acid attaching to the first amino acid via a polypeptide bond. The first tRNA molecule then detaches, leaving behind the amino acid. This process continues until a stop codon codes to stop amino acids from adding to the growing polypeptide chain. The polypeptide chain then undergoes complex folding to shape the protein for it to perform its function. These types of folding include primary structure, secondary structure, tertiary structure, and quaternary structure. Mutations during protein synthesis can arise as a result of mistakes in copying genetic information and can have an effect or be irrelevant. Mutations can arise from substitution, which changes a single base pair by replacing one base for another. Insertions and deletions also cause mutations by adding more bases to the DNA sequence or removing them from a DNA sequence. As codons are read in groups of 3s, this can change the reading of the sequence, changing amino acids.